I prepared a talk today that you saw the title on the last work we've been doing in my team. It's a new team. It was created a little bit more than 2 years ago in IBPS at Sorbonne Université in Paris. But I first want to introduce you to the world of organ-on-chip systems and microphysiological systems. They're basically the same, but you know, organ-on-chip scares a little bit, like a few people, I would say. And microphysiological systems, you will see, is probably a better name because it's also extended to something that's not microfluidics, based on microfluidics. And the aim is to mimic, to imitate what you find in vivo in physiological systems or even pathological systems and try to use those models to better study them or to test drugs on them. So what I will show in the first slides is not our work. I try to place all the references. If you are interested in a particular work, don't hesitate to ask me. I wanted to start with this slide, which is, when I teach, is the last slide I give to the students, but for you it's the first one. I don't know if you're aware of that, but the FDA passed, actually the US Senate passed a new bill starting in January this year. It was actually pushed in September last year. It was called the FDA Modernization Act 2.0, where they say basically now that you can test drugs in non-animal models based on, provided that you can prove that your model is very physiologically relevant, human relevant. So it's stated now in the law that you can use organ-on-chip system and microphysiological systems also to evaluate drug safety and efficacy. So of course, this means that you need to prove that your model is relevant. And you need, of course, microscopy for that. I'm sure you know about that. So organ-on-chip systems, this is the National Geographic cover from January 2019, which helped me explain to my family that I was not Frankenstein in my lab building organs, but basically using this type of chips, which are not microelectronics chips, actually, but they're called like that because they come from the microelectronics world, you know, all the microfabrication tools, which I will not detail today. But basically, you have to think about this new revolution in medicine as a home away from home for the cells. So you basically trick them into believing, sensing the environment like they were at home for different tissues specific to different organs. And for that, it's a synergical work between microtechnology, microelectronics from the beginning, and then biomaterials and so forth, and cell biology. And this is because in brief, mechanically, cells can sense their environment. I think you know about that. We've seen brilliant talks this morning about that also, from the focal adhesions, but also tight junctions, where mechanics is transferred and actually acts as a signal. So you have the mechanosensing and then mechanotransduction. You have pathways that are activated through mechanics. Gene expression is changed in matters of hours or days. I worked with liver cells. You will see a little bit of that afterwards. Hepatocytes change in a few hours in vitro, in culture, because what they sense is actually very stiff compared to the softness of the physiological tissue. Even a pathological tissue is very soft compared to glass. And so cells perceive, respond, and adapt themselves to their microenvironment, not only the soluble biosignals, but also the mechanical biosignals. And this is one of the signals that we were missing in traditional, now not so traditional in vitro systems. And we want to avoid cells to lose their identity in vitro for a better model to be built. And basically, what defines all the markers, like the passport identity of the cells, are a collection of markers that you decide which one you want to look for or look at. And they all depend on the integration by each cell of biochemical signaling, but also geometry, architecture, composition of all the ligands and the junctions, the mechanics surrounding them, and also the dynamics. So it's kind of mechanics, but also that changes in time. And this is very important because cells are basically integrating all that and deciding how they will react to that. And in vitro, you have infinite, uniform, stiff, very stiff, and static cultures. So the cells never look like what you have in a tissue. Well, not never, but they lack a lot of markers. So we needed to change that. And you know probably that in the literature you can find a lot of biomechanical stimuli like the Fleurish stress in endothelial cells, compressive stresses, interstitial fluid flow, hydrostatic pressure, name any you want. And you can add them, you can implement that on an organ-on-chip system, meaning you can try to add one by one, decoupling them or also summing them, adding them up for cells to get all the signal that they have in vivo, recapitulated if you want, or restored in vitro. And those stimuli also, if it's a very dense slide, I'll go back, all those ones you see, all the arrows basically are telling you that something you can control externally. It's an exogenous signal that you can control with the microfluidic pump, for instance, with the stiffness of the gel that you prepare, with the coating of the protein, the ligands that you will use. You can do that, and you can change them over time. So you can actually look at how cells perceive and respond to those signals. So understanding that, and thanks to microfabrication, organ-on-chip systems have started with a very simple, well, not that simple, but look simple, two-channel systems here, where you have basically multicellularity reduced to its basics, which is an epithelial layer, the one that's basically the organ. So it's an organospecific epithelial cell layer, and an endothelial layer separated by a porous membrane that's in between. So I took the example of the lung on a chip, which is the first very successful organ-on-chip system that was made by Harvard researchers. And they use a PDMS porous membrane with pores of 8 microns. So basically cells cannot travel through, epithelial and endothelial cells cannot travel through, but what they secrete, what they produce, will actually diffuse through that barrier. So it's mimicking what you have in an organ. And this was made possible thanks to George Whitesides, which I think, sorry, there's no S at his first name, George Whitesides, who actually is a chemist and made all the PDMS chemistry available to non-chemist people, and that we can use them in those chips now. So it's a transparent silicon, very simple to produce, and to shape with all the resolution that you need. So that's very, very important to use. And basically for the lung-on-chip system, the way that they explain the story of it is that when you have the epithelial monolayer on top of the endothelial monolayer, which are in separated channels with air on that side and a liquid medium, could even be blood on the other one, that you can actually flush some liquid medium inside, you have a better phenotype than when they are in vitro in core culture, even when you sort them, you know, in separated like cell islands. But they were still lacking something, until they realized that those cells are basically stretched in vivo. You have a periodic breathing, and the alveolar sacs are being expanded slightly, but still they're expanded. So they managed physically to create some vacuum to the side chambers here, using pumps also, that are deforming, like when you pull on a straw, you know, and you have your finger on the other hand of the straw, and the straw shrinks. So you create a vacuum here, and when you shrink those channels, the flexible PDMS membrane that's in between will expand. It will actually stretch. You control the vacuum, so you control stretching amplitude, you control the frequency of that also, and they proved in a few papers that the frequency is important and the amplitude is important to get the right pneumocyte phenotype in this chip. So that's very important to have it. And it's the same for the endothelial cells. You need the right amount of shear so that they're happy, like with a physiological condition. But they went even beyond that, and they managed to mimic a disease on a chip, an edema on a chip, for instance, that they could test then for different drugs to look at the efficacy and the safety of drugs. Of course, now they progressed a lot. It's been 20 years, and the desired phenotypes are restored for weeks now in those organs-on-chip systems. That they can also image, of course, with microscopy. There you have really cool videos online where you can even look at E. coli being phagocytes by immune cells. But it's really, really important that you remember that the important part is that the desired phenotypes can be restored on those chips. Okay, so again, I go back to one of the previous slides I had. You can add fluid flow, the right architecture, the right mechanical stimuli, also oxygen gradients. I will not talk about them, but they're important. In this case, you can't use PDMS because it's porous to gases. You need a different plastic where oxygen from the environment will not go inside. And of course, core culture. So this organ-on-chip system is now expanding a lot, and you have even multi-organs on chips. And you probably have read about human on a chip. That's very scary, and that's why we call them now microphysiological systems. We don't want people to think we're building small humans in the lab. But you will, which is actually not true. You will see a lot of examples also. I will focus on this one because it's very impressive what you can get with an organ-on-chip system, but you have the kidney, the endothelium, even blood-brain barrier. And I like this one because I hope some people here have taken Benoit Charlot's workshop where you can actually integrate microelectrodes inside those organ-on-chip systems to either add some stimuli, not mechanical in this case, or to register activity. And in this case, they have a blood-brain barrier, and they address this barrier effect thanks to a tear electric measurement. So everything can be added on those ones, and I will focus on the gut and show you a few approaches. Depending on what you want to ask, depending on the fidelity you need on your model, and of course, your capacity to build them, you have different approaches. But basically, you always have to pick the right chip material. I said PDMS is the main one. It's very simple, but sometimes you need acrylic, as I said, to avoid gases to go through. You need the right membrane, porous membrane. Usually, it's PDMS. It could be polycarbonate. It could be another one. In our case, if you're interested, we modified a protocol that was published by Penn's researchers with collagen 1 that you dry up, basically, and you have like a piece of Kleenex that's very fragile, but you can place in between those two channels, which is more physiologically relevant. Cells can deform it. You pick up the right epithelial cells you want, the endothelial cells you like. Usually, you will see UVEX. They're human umbilical cord vein endothelial cells. And they're very plastic, so they can adapt to the different epithelia that you will have. And you can coat the membrane with whichever protein you want or you need. So you just need to learn the protocols that are published. There is some time that you need to get adapted to those, but it's not that difficult, and that's the first approach that I would recommend that you apply. But you can also guide spontaneous tissue morphogenesis on the chip. So it's not a flat monolayer on the epithelial side, but you have some sort of 2.5D or 3D structure that's actually guided. You don't preform it in advance. You let the cells slightly and slowly, gradually actually organize. And in the gut, it's very impressive. Using CACO2 cells, you compare here CACO2 with organoids, which are known to form 3D structures. And the CACO2 cells with the right amount of shear. And actually in this paper, they show that you also need the peristalsis. So you need the vacuum channels to the side to also have this mechanical stretching. So with the right amount of physiological systems inside the chip, you will see that those cells are forming spontaneously microvilli. And the phenotypes is much, much better than what you would find on a monolayer, even compared to an organ-on-chip system. So now those models are expanding. They managed to get the right phenotype for up to 1 month. So it's very good for drug testing. And of course, you only have epithelial and endothelial cells, and you can add some multicellularity if you want. And you probably have seen this paper. Of course, I like the reference. It's Mathieu HAUTEFEUILLE's paper on the mini guts on the chip, where it's a bit more, well, a lot more complex. And they use some UV laser beam to actually pattern a gel. It's like a developmental extracellular matrix gel, kind of like matrix gel, very rich in laminin. They pattern it to culture the cells, and the cells will actually conform to the shapes that you create here. And you have a flow inside, and they can keep the mini gut on the chip for up to 1 month also. And in this case, it's more for basic research, I would say, and study cancer and how cancer is propagating inside the gut. So it's very impressive. And in this case, it's spontaneous tissue morphogenesis from organoids on a chip. And the fourth approach is what I would call an adaptive design following spontaneous tissue morphogenesis. So you can even change your design while cells are being cultured. And in this case, for instance, you have endothelial cells seated inside an hydrogel. You create, I will switch to this view here, you create a pressure difference between the two lateral channels. So you have an interstitial flow inside the collagen gel here. Imagine a sponge, and you have a flow, very, very small flow going inside. Cells are sensing this flow, they're adapting to it, and they spontaneously create a microvessel that's perfusable. It's actually perfused from this end to the other end. What you see in green here are fibroblasts, which are actually providing the right multicellularity signals for the endothelial cells to build the microvessels. So you have the microvessels formed inside the gel here in 3D, as you can see from this perspective. And you can add then, after your organ-on-chip system has managed to produce with spontaneous tissue morphogenesis the right capillary bed, as we call it, you can add some tumor organoid also. It's very tricky with organoids. We actually discussed that last week with a colleague of mine, and they said it's very difficult to vascularize organoids, but with tumor it works well. And you promote anastomosis with a spheroid or a tumor, and hopefully in the future with organoids. So you can adapt your design as time passes and ask the questions that you want. So now there's a lot to discuss about all that. I'm guessing you already have a lot of ideas and how you can image all those. But what I want you to focus on now is what I'm interested in, is a lot of things are common in these models. 2 channels, epithelial, endothelial, porous membrane, different types of cells. But what's actually very important here are the endothelial cells. What made the difference in the first example of the lung on a chip, you remember the first I showed you, is adding this monolayer of the endothelial cells. Because you already had a monolayer of epithelial cells, you could already add mechanically the stretching if you like, but if you don't have their neighbors, the endothelial cells, it doesn't work. And why is that? Because it's part of the basic element of the organ-on-chip system, because traffic is regulated by them. So what goes through this membrane is actually, this passive membrane is actively regulated by the endothelial cells. It's been proven. You can control the flow, so you can control also the conditions under which your cells are cultured and how the phenotype will change. You can, for instance, stress them that much, like you have an inflammatory phenotype, and look at how they affect the epithelial cells. You can look at the immune cells migration through that barrier also, and cytokine transports. And that led to a very nice example that was published last year in 2022, organized around Gordana Vunjak-Novakovic, who was a pioneer in organ-on-chip systems. It's a multi-organ-on-chip for drug testing, where they want to look at the possible effect of a drug on the liver, the heart, skin, if it's a cream, for instance, and bone marrow. So they picked 4 groups who already had their own organ-on-chip system that was working, was published, worked well, 2 channels for each. And they decided to interconnect them. And you know, when you start working with multi-organ-on-chip system, the easy way to go is you take the supernatant from one organ and you place it in the next organ and you look at they interact. But it doesn't work. It doesn't work because endothelial cells are the regulator of what goes through, in and out. So they had this idea of comparing individual organ-on-chip system, which worked quite well, isolated, compared to the adults. Here you see in the genes, they look quite close to the organ you want to imitate. And when they mix them with the lower channel here and just the porous membrane, it degraded a lot the phenotype that you expected for the in vivo recapitulation of the in vitro model. And that's because you need the endothelial barrier here. They are doing something which was not actually shown very in detail in that paper, but they are doing something in that regulation, and it's always the winner in the transported media versus the shared media. So one of the questions you may have is, when you do multicellular culture, how do you deal with the different media that you need? You know, epithelial cells don't need the same medium as the endothelial cells. Well, those guys actually regulate all that, and you can pretty much use a transported medium between them. So they are building now. You see how it looks like. This is the engineered multi-organ-on-chip system that will go into industry. It was actually paid by industry, and they will test the drugs there. But you can think about fundamental basic questions also, and this was a question that Donald Ingber, another pioneer of the organ-on-chip system, is the one who actually made the lung-on-a-chip possible. And he asked this question: is it time for a reviewer 3 to request human organ-on-chip models instead of animal validation? Organoids or organ-on-chip. And that's because they are more physiologically relevant. They preserve for longer the phenotype that you want, pathological or physiological. And you can do some mechanistic studies inside, and that's what we're interested in. And also drug testing, why not? But that's why the FDA was convinced. They actually paid for those studies 10 to 15 years ago, many groups in the US, and now it's something that becomes real. So I will now focus on the work that we are doing in my team. So before I came to Sorbonne, I had a team in Mexico City, as you said. For 10 years, we focused on a liver organ-on-chip system. It's something that's been made already, but it doesn't work quite well. You don't find the right phenotype, especially if you want to study fibrosis. This is why I asked about the collagen formation. So I spent almost 10 years to try to have the hepatocytes happy inside an organ-on-chip system as a monolayer. They really transdifferentiate fast. And long story very short, we use the soft hydrogel coated with collagen 1, and we can preserve them for between 5 to 7 days. But they were lacking something. And you ask the physiologists, you ask the medical doctors, and they say, of course, they need a lot of signals from the endothelial cells. And that's the lesson we have from organ-on-chip system. And this is a drawing from a lot of years ago when we had this idea of using this porous membrane and endothelial cells from the liver, they're called LSECs. And it never worked. They actually got worse, which is exactly the contrary of what I showed you before. So what do you do? We realized that some papers were saying that in certain types of capillaries, like the liver, but also the brain-blood barrier, monolayers are weak because they are very dense. So the barrier function is actually blocking too much. And they can become leaky afterwards also. So that's perturbation for the hepatocytes. And the reason why is because it's known now that the curvature is one of the signals that those cells need to behave as liver endothelial cells. So we decided to see if it was possible to engineer a tube of endothelial cells and then have this co-culture thanks to a tube. And that was the work of Jimena, a PhD student from Mexico who joined me in Paris, actually. She came here for almost 2 years. We just submitted our work to bioRxiv, so 10 years bioRxiv, 20 years Mifobio, I think it's signed. And I will try to be quick because I know you want to go for lunch. I just want to show you a little bit about the liver. So it's a very big organ. It's one of the most vascularized organ in your body. It can regenerate if we cut down to or up to 3/4 of your liver. It will regenerate spontaneously in 2 weeks. You can transplant a very small liver to a receiver, and it will grow, or it will shrink if it's too big, in less than 2 weeks. And it's organized in lobules, which we published some work on that, showing that the hexagonal shape has a function, as a vasculature. And the blood flows basically from the outer points to the central vein here, inside what we call sinusoids. So they're not straight like that, they're sinusoidal in 3D. It's very difficult to draw. So we simplify by showing you it's a blood vessel like this, a capillary. But it's discontinuous, so it's not tight. Monolayer, very, very far from what we want. And there's no basement membrane, almost no basement membrane, which is very weird for a capillary. Actually, the hepatologist will tell you it's the only one. And then you talk to a neurologist and they say, no, it's not the only one. But it's zonated also, the phenotype changes. And this is only, in human, is less than 1 millimeter in length. So the identity of those endothelial cells will change along this. So that's something we would like to restore at some point. But let's start with what they do in the liver. And usually what you know about liver fibrosis is that you have some cells here, hepatic stellate cells that are quiescent, and when you need to repair, they produce a lot of ECM, mainly collagen 1, which is good for repairing, but when they go crazy, they just never stop, and you get a stiffer liver, and you get fibrosis, probably cirrhosis if it continues, if it's chronic, and even cancer. And it was thought for a long time that they were the ones, the only culprits. And now we know, thanks to in vivo models, that the LSECs here, the ones that are making these tubes, the microcapillaries, are probably the starter of all that because they sense a change in blood flow. Like hypertension, for instance, will increase the luminal pressure, the shear stress, and it will signal to those cells, and those cells will then trigger what we thought were the culprits. So they were told to produce ECM, and nobody knows why. And we don't know why because we need a model to do that. And that's why we want to build that model. So if we need a tubular system, we looked in the literature, we talked with colleagues and friends like Claire Dessalles, for instance, from Polytechnique also, in Abdul Barakat's team, where they were making a tubular structure for endothelial cells. So basically you have a collagen gel here, but while you're gelating, you place an acupuncture needle, you can choose the caliber, you gelate, and then you take the needle out. So you have, imagine a spaghetti inside the gel that you make, or the jello in your fridge. You take the spaghetti out and you have a tube, an empty tube. And then you seed the cells, you coat basically the walls with endothelial cells, and you have a tube. So we tried that, but unfortunately, endothelial cells in the liver don't want collagen around them. They change their phenotype straight away. So not a good idea. We should have known that before. We also tested the example I showed you before about the capillary bed. We placed some L-sex inside the gel. We placed a pressure difference on both sides. They organize spontaneously inside the microvessels, but again, they are trapped. They're embedded in a gel. We cannot remove the gel afterwards. It won't work. So it's not a good idea. So we need a model where the tube is actually self-sustained and not embedded in gel. So we went to development. How do endothelial cells form tubes in development? They're not embedded all the time, especially in the liver. So usually you have, that's the model that you will find in books, you have vasculogenesis, so progenitor cells will end up forming cords and eventually a tube with a lumen. So lumenless cords, organization, and then vessels are formed. There's something that's close to that in vitro, which is called the vasculogenesis assay. You use a matrigel, a thin layer of matrigel, even a coating. You place endothelial cells on top. It works with a lot of different types of endothelial cells. We tested with the different cell lines and even rat primary and hepatic endothelial cells. And they all form these nice, you see it's a complete petri dish. These nice lumenless cords. A lot of people you will see in the literature call them tubes. We haven't seen any lumen, at least in the hepatic cells. So you need to remember here that soft laminin gel is necessary, and 2 papers were published just during COVID, showing that it's a mechanical issue. So cells are pulling on the laminin. Laminin is more plastic than collagen. Collagen is elastic. You know, as you release the force, it will go back to its original shape. Laminin, at least it's a very high relaxation time, but it seems like it's plastic. It stays in its position. So it's helping cells to recognize each other, to migrate towards each other, and to form those networks. But no lumen is formed, and after some time it even disappears, it gets thinner. And I wanted to show you here that it depends on the stiffness, you know, the island and the dimensions of those structures. And also, it's random. You don't structure laminin, it's not fibrous, it's organized very randomly. So how can you try to form a long-lasted structure that you can wait or expect that will form a lumen after some time? It needs time, it needs flow. How do you do that? So the first idea that we had is, can we organize the laminin for a preferred direction and some anisotropy? Is it possible to do that, and then the cells will form a train and maybe a longer lasted structure? And we found a really nice paper from Celeste Nelson, who used sessile ECM droplet. She used collagen 1. Actually, I can show you now. She used collagen 1, and she controlled the evaporation rate versus the gelation time. And by doing that, you have what we call a Marangoni effect. I don't know if you know that, but you probably know the coffee stain effect. You know, when coffee dries, all the coffee microparticles will stay on the outer edge of the drop. So the drop will actually dry out, but it ends up being more colored on the outside of the drop. Do you understand that? It's called the Marangoni flow because of the evaporation flow that you have here. So it's concentrating a lot of particles here. And what Celeste did was actually very clever. They did that with self-assembling collagen fibers, and you can see here at the middle, they're random. Near the edge, they're actually drawn by the Marangoni flow and all organized in one direction, radially outwards. And on the edge, you have a really dense, not really organized, but very dense concentration of collagen fibers. And we did exactly, well, Jimena did exactly that with laminin. Can we do this with laminin? So we controlled the gelation time. We didn't have matrigel after COVID. I think you know about that. So we bought Geltrex. It worked well in our hands. She found the right conditions for the evaporation and gelation control to get a middle region near edge and edge, very dense here. So you don't see any fibers. It's normal. It's not collagen. It's more mainly laminin, not only, but mainly laminin. And when she labeled laminin and collagen 4, which is another ECM component of the Trex and matrigel, you see they really densify towards the edge. You see here even the filling factor is greater. So we took those droplets. It doesn't really depend on the size of the droplets, by the way. down to a certain size, of course, but it doesn't really depend on that. But it depends a lot on the humidity you have around. So we bought a humidity sensor, and now we control all that. And then we culture cells on that. So all you will see now in the next slides are for a cell line of hepatic endothelial cells called T-secs. So you plate them. You have the sessile droplet here. You can see the edge very, very clearly. And after 24 hours, we saw the vasculogenesis in the center where it's random. Expected, that's what you find on a monolayer. You have on the gel, sorry. You have a monolayer of cells forming on the glass. They actually like the glass, and we found out that those cells are capable of producing their own laminin, so they spit laminin basically, and they form the monolayer. And we realized that at the edge, something interesting was forming. You see here, like an anisotropic edge form was created. And after some times, we could even see, it looked like a tube. At the center, it's lost progressively, as you would find in any other structure. Jimena did some essays, and during one of the lockdowns, I don't remember which one, she had to leave that for several days, and it remained for up to 27 days at some point, and even without changing the medium for 5 days once. So they're very resilient. I'm not saying they're really happy or in the right shape, but the structure is still there. That's what I want to say. It's not photonic microscopy I'm showing now, it's electronic microscopy, but it tells you a lot about the morphology and the physiology of the cells that you have. The monolayer, of course, you have some cracks in the preparation. Sorry about that. We just learned how to do that, how to prepare our samples. But you can guess where the cells are. And if you go closer, this is 1 micron, you see a lot of philopodia. So they're typical of a monolayer, and they are not always well connected. On the core, do you remember, at the center where you have the vasculogenesis, you have very straight structures, and they look very tight, but they are not really well interconnected. We were happy to find fenestration, which is a good sign for endothelial cells of the liver. This is after 5 days. In monolayer, they lose their fenestration in 24 hours. That's a good sign, but they are still not mature. You would expect a lumen there, but it's not. It's actually a multicellular, like tiles on top of each other. You have no lumen. And this is after 6 days around, so the dome is here. Now it's flat after the preparation for the electron microscopy. You have the monolayer to the side, and separated from both the dome and the monolayer, you have this nice structure made of cells. And you can guess here some fibers aligned in the direction of the anisotropic tube. And this, you will see, was not there 1 or 2 days before that. We tried to understand how those tubes were formed. So thanks to René Marc-Mege and Benoît Ladou's team, they helped us a lot in getting a biostation where we could follow, we could image for a longer time what cells were doing. And after you observe that, you do the confocal microscopy at different time points. And that's exactly what we did. So I will go fast on that. First, it's an apoptosis. If you're interested in the details, you can read the paper. If you want, I can send it to you. It's more an apoptosis. Only the cells that are less than 50 micron from the edge will migrate towards it in any direction, and then they will stay. They will never move away from the edge. They like laminin, and they stay on it. They migrate and proliferate along the edge, and in 1 day, you have all the edge covered. And that's very impressive, because even the larger domes were covered in less than 1 day. And then they organize, as you can see here, like a train, exactly the picture I showed you. And if you look at the transversal view from the side here, you can see that they climb the dome up to 2 or 3 cells high. And that's very important when we realize that, because that's where they climb here. So I thank the photonic microscopy facility we have at IBPS. They helped us a lot in getting those nice pictures. And on stage 3, you see now it's not 1 cell train, it's a multiple 2 to 3 cell train. And if you look in the transversal view, they're actually folding the dome. So they were climbing on the dome. This will be very useful. They're climbing on the dome and they're folding it. You see here. And after some time, we realized that we could measure the angle, so that's the quantification. The folding is progressive. We could realize that we have some depletion of it seems to be that you have a depletion of laminin here where the hinge cell is, exactly where you have these movements at this cell here. We need 3 hands to understand. So progressive folding, you see the angle here, and local ECM depletion. We thought it was probably an apical constriction. We looked for actin a lot. We spent a lot, well, Jimena spent a lot of time looking for it. We never found it. What we think happens now, and we need proof of that, is that it looks like a lateral pulling. If you take your clothes, you know, to clean your glasses, and you pull slightly to the sides, you will see it buckles and it folds downwards. And we actually checked with some fluorescent beads inside the gel, and you do have that lateral pulling first and then folding, which eventually looks like this. It's completely folding over. This is a nice picture after it's completely wrapped. So it's like a blanket that the cells will pull over them, and they're all covered with laminin after some time. And this is very important because in development, that's exactly what happens. They are completely rounded by laminin, and laminin is a signal to polarize apicobasally. So we looked at podocalexin, which is an apical marker of endothelial cells. When you have a lumen, it's because podocalexin was transported from the basal side to the apical side. So I hope it's easy for you to realize what I'm imaging here. I'm going through that tube. So it's like a wave. You see it from the side here. And here you're on the shore and you see the wave coming at you. And you see podocalexin is basal, where the laminin is. And at stage 4, it becomes apical. So it becomes like at the center of the cell-cell junctions here. Of course, we need improvement of these images, but we are starting on that. And now that we have this polarization changing at stage 4, after that, we saw that Z01 was actually maturing at this structure. I think I have a slide on that. I will show it afterwards. And you have a lumen opening at stage 4. So this is a little bit more messy, but inside that, you have a lumen opening. And the lumen will not be sustained if you have nothing flowing inside. So it's sustained for 2 to 3 days, but we need now to perfuse it. And you can look at the side of it. It's very difficult to perfuse. So we are also working on that at the moment. So this is Z01. It's looking like intracellular condensates at stage 3. And the same structure after stage 4, you know, after the podocalexin went apical and you have a lumen forming, you see Z01 going more at the cell-cell junctions. And what's also very interesting is after the blanket was wrapped around the cells, you know, to form this tube, you see it looks like the dome structure. It looks like the laminin and collagen 4 structure. But one day after that, they formed those wavy bundles. We were very surprised by that. We were wondering where it was coming from, maybe the preparation of the samples, but we saw that a lot at the later stages. And this is actually a line in the direction of the tubes. And when we use photonic microscopy, we checked it was laminin and collagen 4 also, but it was produced by the cells. It was not exogenous. So they're maturing their basement membrane, which is kind of bad for the liver. We need to measure the thickness. It shouldn't be there if it's a healthy liver, but maybe nothing is perfusing inside, so by perfusing we could change that. But that's very interesting to see that they are organizing themselves already. So this is the summary of what Jimena drawn to explain what happens: aptotaxis, cord formation as a train, dome deformation, and this wave structure, this folding, that will drive apicobasal polarization, a lumen opening, and lumen formation that we now need to perfuse to sustain for a longer time. And then came Wenjin. She actually helped a lot in imaging all that. Wenjin is a postdoc researcher in my team. She joined last year. She's very, very good with proposing new ideas and trying to go beyond that and to perfuse. She thought maybe a circular shape of a vessel is not that good. Let's try a straight vessel. So she tried different strategies, using gel tracks all the time, aligning droplets, like imagine you are the bioprinter and you place the droplets and they coalesce. But unfortunately, you see here, you still have a shape of droplets like this, but it's still a straight tube and it's forming exactly the same way. She also tried a hanging strip. So she made a strip and then made it hang. So you have this dome shape again. And in this case, it's even more straight, and they also form those tubes. And now she's working on a PDMS support also, because now we want to anastomose those with collagen gel and try to perfuse by coaling the cells inside the gel, you know, like the vasculogenesis inside the gel. And it also works around the PDMS support. This is what the structure looked like around the PDMS and gel tracks. You can see a different structure. And what's very impressive, and we still don't really understand why, they go much faster. It took 4 to 5 days in the circular shape. It takes only 2 days on a straight train. So that's very impressive. And now we are working with interstitial flow. I will finish with a few slides only to show that we are trying to have a flow on top of them and maybe have the flow going inside, providing that at some point they are not very tightly connected, to go from discontinuous to fenestrated phenotype. We'll see if it works. And we're also trying to anastomose the droplet that we have here with the collagen gel, because we managed to get some angiogenesis under VEGF chemotaxis, if you like. So cells are going inside the gel, and hopefully we can perfuse through that. But we are also benefiting from the fact that we are in developmental biology lab and try to anastomose with chicken embryos vasculature. So here you see the glass with the dome somewhere here that is trying to anastomose with our vessel. And what we found, it's very preliminary. We did only once, but there are our cells here. We use human cells in this case. Doesn't work with mouse cell lines. So with human cells, these ones are the ones sprouting towards the vessels of the chicken embryo. Usually you have the other way around. The chicken embryo vasculature is trying to vascularize what you put in contact with it. So I finished with that. I'm very proud of starting this team at Sorbonne University. I thank all my teammates, only students and postdocs now. All my collaborators who helped me a lot in developing that, and they helped Jimena a lot also, so much so that she wants to come back to France for a postdoc. And of course, I thank you for inviting me. And it's my first Mifobio. I think it will not be the last. And you for your attention. And one last message. I just knew 2 days ago that we can recruit an ingénieur d'études between my team and the microfabrication and microscopy platform. So if you're interested, let me know. Thank you.
Thanks, Mathieu, for the nice talk and overview. I was wondering what finally happened to the laminin. So do the cell detach the laminin from the
cord?
Yeah.
No, it's very tightly attached. Actually, the monolayer detaches when you do some sample preparations. The cords at the center, they detach very easily. Sometimes they even form spheroids and they detach, but the tube around is very tightly attached to the gel dregs, very tightly.
Okay, and when you have this image of the blanket being folded around the tube, is it also detaching? So the blanket is the laminin, right?
Yeah, no, it's not. It's not detaching. It's pulled. It's pulled from the sides here. First, it is half an hour later, and then here it goes inwards. So it's never detaching. It's like really they're pulling on the. It took us a long time to believe what we were seeing. We saw a front going outwards like this, like you would pull piecewise, and that's where it's actually falling, and with the cells that's here falling on top. So you have a tube forming like a wave, basically. It's exactly like a wave, but it's never detaching from the rest. It has to be very soft. When you don't control the gelation very carefully, and we measured it with a nano indenter, only 1 kilopascal above that, and it doesn't fold anymore. They form a monolayer on top. It has to be very soft. So it's a combination of laminin and softness. Thank you for your question.
Excuse me, just before we take another question, I wanted to let you know that there will be the big group picture right away. So if you leave the room, don't forget to go to the pétanque area just nearby where the fire alarms took you the other day for the big group picture, and spread the word for the ones who are not aware. So if you have colleagues already in the canteen, let them know, and then we can go on with the question if you have any others. Thanks.
Yeah, thank you for the talk. I have a question maybe more related to the first part of this microfluidic device, although the second part is also very, very, very beautiful. So you now have this microfluidic device. I'm just wondering, how are you going to image through the microfluidic device? Of course, that depends also on the resolution you need. So let's imagine I want high resolution. I probably want to use a high N8 objective lens, and then the working distance might be the problem. And sometimes I understand that you might also have an error on the top of the cells, so you cannot use the water immerse objective. So how do you overcome with that problem?
Yeah, it's a very good question. And it depends a lot on the application you have in the chip design. So basically you design your chip depending on the questions you want to make. Sometimes you want to do RNA-seq, so you don't care about imaging and you do a thick chip and to have a lot of volume that you can take out afterwards. But if you want to image, you basically create your chip on top of a very thin glass and you image from the bottom. And but what we found was really good is actually an air objective with a large field of view, but you can still go inside the thickness to have all the tissue you want. If you want precision resolution, then you can flip the chip, you can image from different directions. And there are a few papers, quite a few actually, who explain, depending on the question you have biologically, how you can image. And how to make your design according to what you want to image and how you want to image. So I don't have a straight answer, it depends. In engineering, it's always the answer. It depends. And you can look in the literature and I'm sure you'll find what you want. Otherwise, the good thing is to go with your platform engineers and ask them, I want to do this and what do we need to do? And it usually works. Thank you.
More questions? I will try last one.
Yeah.
So for the future, so how do you think you can perfuse your tube?
So ask Wenjin. It's very tricky. We first try to poke at it, basically, using very thin capillaries and with a pump. We found that it's possible, but they break out. It's too aggressive. So the best guess we have now is to actually do like the angiogenesis assay. So you can have an interstitial flow going from one side to the other. And inside this collagen gel here, you will place a VEGF-rich medium. So you create a gradient inside the gel and you will attract some angiogenic sprouts from the tube towards the other end. And because it works in a chip and it's completely perfusable, we hope that we can interconnect our pump with the capillary to those opening vessels, new vessels that are going sprouting from our tube inside the vessel. That's one of our possibilities. The other one is through anastomosis. A lot of tumor vascularization or organoid vascularization happens in this kind of model. And we just started, we'll see if it works. Then you cut around and you take everything with the vessels.
Okay.
That's the idea.